Hello and welcome to video 4 in the endocrine system for anatomy and physiology. We are going to be in the diagram packet on page 4, and we're going to look at this type of membrane-bound receptor called a G protein. And this is something that many membrane-bound receptors work by activating G proteins. And this G protein is named because attached to it is something called guanine diphosphate. And guanine, if you remember, is one of the 4 nucleotides. This is the G in the ACGT DNA nucleotides. So we have a molecule guanine with 2 phosphates attached, and it's sometimes called GDP. So this G protein actually has 3 subunits, and they are given Greek letters alpha, beta, and gamma. And on the alpha protein, as you can see, we have a molecule of GDP. So in the first diagram over here, we have our receptor, and it's membrane bound, so it spans the membrane. And we have a water-soluble hormone that can attach to this receptor. So, excuse me, the hormone is not bound to the receptor. In this first diagram, GDP is attached to the alpha subunit on G protein. So this is before anything gets started. Then we're going to move over to the second diagram. So we have the water-soluble hormone binding to the receptor. And then you can see how the diagram changes. So the hormone binds to the receptor, which we know if something binds to a receptor, we get a change in shape. And the G protein actually moves and attaches to the receptor. That causes GDP to be replaced by GTP, and you can probably guess that stands for guanine triphosphate, so it phosphorylates it. It's actually just a switch. So the process of the G protein binding to the receptor causes a change in shape, and the GDP falls off. GTP, which has an affinity for binding to that site and is in much greater quantity, automatically fills in that space. And this activates the G protein. So moving down to diagram number 3, which is here, we still have the hormone bound to the receptor. Because we activated the G protein by replacing GDP with GTP, that causes the G protein to separate, which then the alpha subunit separates from the other. And what happens here is this causes the cellular response. Excuse me. So as this GTP-bound alpha subunit separates from the other subunits, we get a cellular response. And these can include a lot of other things. These can include ion channels, open or close. We could have secondary messengers. We could have enzyme activation. And we're going to look at one of those in the next video, and that's going to be enzyme activation. But after this happens and we start the cellular response, we have to go back to normal. So if we look what's happening in this next diagram, The water-soluble hormone separates. Well, that causes another change in shape. The phosphate is removed from GDP, which turns it into GDP, and we go back to step 1. So whenever this phosphate is removed, this alpha subunit is going to move back and attach to the beta subunit, and we're going to be the whole way back here to step 1. So basically, this is almost like a conveyor belt sort of thing. You have something bind, it causes changes, those changes then cause more changes, and then we revert back to the same thing. So we've seen this over and over and over again in the body. This is just specifically for water-soluble hormones, and there are a lot of different hormones that are activated or activate cellular processes through G protein. So the next video is going to be on the activation of an enzyme by G protein.